In this lecture, we will learn about citric acid cycle or tricarboxylic acid cycle or Krebs cycle, as it was first proposed by Adolf Krebs in 1937. It is the final oxidative pathway that completely oxidizes acetyl-CoA to carbon dioxide. It is amphibolic in nature, that is, helps in the making and breaking of the molecules. Pyruvate derived from glycolysis is oxidatively decarboxylated to acetyl-CoA by the pyruvate dehydrogenase complex. All the enzymes of the citric acid cycle are located inside the mitochondria except succinate dehydrogenase, which is partially embedded in the inner mitochondrial membrane. Oxaloacetate acts as a catalyst which enters in the reaction, causes complete oxidation of acetyl-CoA to carbon dioxide and comes out of it without any change. Oxaloacetate is an important junction point in metabolism. TCA cycle integrates all the major metabolic pathways. TCA cycle also shows anaplerotic role, that is the replenishing of the cycle's intermediates. Now let us see the citric acid cycle in detail. Pyruvate, which is derived from the glycolysis, gets converted to acetyl-CoA. Acetyl-CoA along with oxaloacetate forms citrate, which is a 6-carbon compound. This citrate in the presence of aconitase releases a molecule of water and gives cis-aconitate. Cis-aconitate in the presence of aconitase forms isocitrate. Isocitrate in the presence of isocitrate dehydrogenase reduces a molecule of NAD to NADH plus H plus and forms oxalosuccinate. Oxalosuccinate in the presence of isocitrate dehydrogenase releases a molecule of carbon dioxide and forms alpha ketoglutarate, which is a 5-carbon compound. Alpha ketoglutarate in the presence of alpha ketoglutarate dehydrogenase releases a carbon dioxide molecule and reduces NAD to NADH plus H plus and forms succinyl-CoA. Succinyl-CoA is a 4-carbon compound. Succinyl-CoA in the presence of succinate thiokinase gives succinate and a molecule of GDP gets converted to GTP. Succinate in the presence of succinate dehydrogenase gives fumarate. Here a molecule of FAD gets reduced to FADH2. This step can be inhibited through malonate. Fumarate in the presence of fumarase takes in a water molecule and gives L-malate, which is also a 4-carbon compound. L-malate in the presence of malate dehydrogenase gives oxaloacetate and a molecule of NAD plus gets reduced to NADH. This oxaloacetate along with acetyl-CoA This oxaloacetate along with acetyl-CoA forms citrate and can perform one more round of TCA cycle. Now let us see the energetics of the TCA cycle. Overall, 3 NADH and 1 FADH2 and 1 molecule of GTP are produced from one round of TCA cycle. That is 1 NADH from in this step isocitrate to oxalosuccinate, and the other NADH is produced in this step alpha ketoglutarate to succinyl-CoA, and the other NADH is produced from malate to oxaloacetate. FADH2 is produced in this step succinate to fumarate. GTP is produced from this step succinyl-CoA to succinate. So overall 3 NADH, 1 FADH2 and 1 molecule of GTP are released from one cycle of TCA cycle. Each NADH molecule produces 2.5 ATP and each FADH2 molecule produces 1.5 ATP and one molecule of GTP releases 1 ATP. That is, overall 10 ATP molecules are synthesized from each cycle. Each molecule of glucose releases 2 pyruvate molecules through glycolysis, and each pyruvate can produce 10 ATP from one cycle of TCA cycle. That is, from one glucose molecule we can obtain 20 molecules of ATP. Though ATP is not synthesized directly from the TCA cycle through NADH, FADH2 and GTP, it can synthesize ATP through electron transport chain. As we have previously discussed, the TCA cycle is amphibolic in nature. Here we can see citrate gets converted to fatty acids and cholesterol, and alpha ketoglutarate participates in amino acid degradation and synthesis, and succinyl CoA helps in the formation of porphyrins, and isoleucine, methionine, valine on odd chain fatty acids gets converted to succinyl CoA, and aspartate, phenylalanine and tyrosine amino acids gets converted to fumarate in their degradative pathways, and malate gets converted to glucose through malate shuttle that we will discuss in some other video, and oxaloacetate gets converted to a few amino acids, and oxaloacetate can be synthesized as well from amino acids, and carbon dioxide is released through this cycle. Thank you.